Welcome to our research lab dedicated to the study of dairy. My name is Joana Ortega and I work as a postdoctoral researcher in the group. In this tour, I'm going to show you our capabilities and the tools that we use every day in the study of dairy science. We have two facilities with the resources to produce and analyze every dairy product. One is in Parker Building, and the other is in Howland Hall. When it comes to processing, and thanks to Dr. Alvarez, we have access to a benchtop homogenizer with two stages. We also own a membrane filtration system with cassette membranes that range between 3 and 50 kilodalton molecular weight cutoff. In addition, we also have a carbon dioxide supercritical fluid extraction system to perform green chemistry when it comes to the extraction of fats and other hydrophobic compounds in milk and dairy. To analyze our products and samples, we have fast instruments with optimized methodologies to perform compositional analysis. The Smart 6 is a microwave and infrared moisture and solids analyzer, and the Smart Track 2 is a module that uses a low-resolution NMR for fat analysis. The protein analyzer is based in a rapid calorimetric reaction to avoid production of toxic wastes. In addition, we have a nitrogen analyzer located in Howland Hall, used to determine total protein content via an optimized Duma combustion. This means we don't generate toxic waste versus other methodologies. When it comes to analytical techniques, firstly, we are capable of performing any spectrophotometric analysis. Moreover, we do protein analysis by gel electrophoresis and high-performance liquid chromatography, which we also apply to the quantitation of milk phospholipids. We also have everything necessary to perform microbiology and molecular biology experiments, usually with lactic acid bacteria and milk proteins with a potential application in the industry. We have the basics to perform optical microscopy. However, a special mention has to go to our state-of-the-art 3D laser microscope that allows us to analyze surfaces in 3 dimensions and determine surface parameters such as roughness, area, and volume. We are also very proud of our instrumentation for physicochemistry, starting with our dynamic light scattering to characterize particle size and charge in colloidal solutions such as milk and milk derivatives. A more specialized instrument like our quartz crystal microbalance allows us to study molecular interactions in real time, like the formation of biofilms upon heat treatment, protein denaturation, or even interaction of probiotics with milk components. We currently own the flow module, the ellipsometry module that should connect to an ellipsometer to study conformational changes in polymers and proteins, the open window module that can be attached to the microscope and obtain interaction images in real time, and the high temperature, high pressure module that allows us to simulate UHT conditions. Last but not least, our research group owns a cell culture lab located in Howland Hall, when we can cultivate various cell lines, including human intestinal cells such as CACO2 and CACO2H29 co-cultures, mouse adipocytes, and more recently, human keratinocytes. In this lab, we are able to determine the change in cell morphology, biochemistry, and gene expression upon treatments that induce inflammation, translocation, and cell survival due to the addition of lactic acid bacteria, milk phospholipids, and other milk components. In conclusion, in our lab we have everything necessary to do world-renowned dairy science with the best tools available.